Okay, so we're going to talk about the pentose phosphate pathway and fermentation. There's not too much about it on the MCAT, but it is absolutely fair game, so we're definitely going to go over it. So the pentose phosphate pathway is also known as the PPP or the pentose phosphate shunt. This pathway is essential to nucleic acid synthesis and other anabolic processes. So it's really simple. Basically, we're starting off the same exact way as glycolysis. We have glucose. If you look right over here to the right at this diagram, we're starting with glucose. HK, that stands for that hexokinase, that first irreversible step. We create G6P. So if you remember, this next step would be the committed step if we were following the glycolytic pathway, but there's also another option. It can enter the pentose phosphate pathway. So this G6P right here at step 2 can sort of take a sidestep into the pentose phosphate pathway if necessary. So the pentose phosphate pathway, there are 3 important molecules that are produced. So NADPH is probably one of the most important ones, and it's useful during anabolism. NADPH, if you see that H, you know it's a reduced cofactor. So we have NADH, we also have NADPH. So this is where the majority of NADPH in your body is created. And that molecule is pretty useful during anabolism, specifically with fatty acid synthesis, we see it used. Another product of the phosphate pathway is ribose 5-phosphate, which is a phosphorylated sugar that can be used to create nucleotides. Any nucleotides that have ribose in their backbone, that's how we create that as well. And then it will also come out with glycolytic intermediates that will feed back into glycolysis. So if we're looking back over here, this green and purple phase is the pentose phosphate pathway. And then we get right over here to the left of this purple, and it's going to feed right back into the glycolytic pathway. And that's really all you need to know about the pentose phosphate pathway. It's just sort of another, another energetic pathway that we use to create important biomolecules like NADPH, ribose 5-phosphate, and glycolytic. Just the name of the actual glycolytic intermediates aren't really important, but it's just important to understand that that's why we call this a shunt, because we could pull reactants out of the glycolytic pathway and we can shunt them back in halfway down the line so that we don't really waste anything. So fermentation, there are 2 types of fermentation. There's lactic acid fermentation and ethanol fermentation. In humans, it is lactic acid, that is the byproduct. So in anaerobic conditions, the electron transport chain and oxidative phosphorylation cannot function properly. So oxygen is the final electron acceptor in the electron transport chain, if you remember. Without anywhere for the electrons to go, we see a buildup of NADH and a halt of the ETC. So all of that, those high energy electron carriers, all that NADH that we're making from glycolysis and the Krebs cycle, if there's nowhere for it to dump its electrons, it's pretty much it's just going to mess up the delicate balance of the entire system. So we're going to have a buildup of NADH and we're not going to have any NAD+. So this becomes not only an issue for the electron transport chain, we're not actually performing oxidative phosphorylation anymore, but it messes up the preceding steps as well. So the supply of NAD+ has been completely reduced to NADH. Glycolysis may be inhibited because reduction of NAD+ to NADH is a key step and acts as an oxidizing agent, a very important oxidizing agent in that process. So if the body could replenish its oxidized NAD+ levels, glycolysis can continue in their anaerobic conditions. So pretty much the most intuitive way that the body thought of replenishing NAD+ levels from NADH is if you don't have oxygen, you just have to find a different final electron acceptor. So that's exactly what the body does. And it turns out that that electron acceptor is actually pyruvate. So during fermentation, the body finds another electron acceptor. When pyruvate receives electrons, NADH is oxidized to NAD+, so we have that reduced NADH form. Instead of giving it to oxygen like it usually does during the electron transport chain, if there is no oxygen present in the environment, it will donate its electrons to pyruvate, and that's how it's going to regenerate that oxidized form of NAD+, and pyruvate is reduced to lactic acid. So in certain organisms like yeast, pyruvate is reduced to ethanol instead of lactic acid. So those are the 2 main forms of fermentation, but that is really fermentation's goal. It's not necessarily to create ATP, but it's to replenish those NAD+ molecules so that we can get through glycolysis and we can create ATP in other ways. Ethanol and lactic acid are toxic at high concentrations, so fermentation has a limit. It's not something that should be performed in ideal conditions, and it's not something that's performed in ideal conditions. However, the buildup of lactic acid, our body has figured out sort of a clever way to recycle it and create energy from that lactic acid, and that's what we refer to as the Cori cycle. So the Cori cycle is a way for the body to reuse the lactic acid created during fermentation. So when oxygen supplies return to normal and we finally have oxygen back into the blood, oxygen supplied to our muscles, the body is also going to start replenishing its NAD+, right? So as we have those NADH, those reducing agents, we finally have oxygen for those electrons to be given to. So we're finally turning all that NADH back into NAD+, and the ETC is going to return to normal functioning. So as it returns to normal function, your body is trying to get rid of lactic acid, so it's going to send it to the liver, which is, you know, the part of your body that takes care of purification of toxins throughout your blood. So it's pretty intuitive that it would end up in the liver. And during the Cori cycle, lactate is oxidized back into pyruvate. So we're just taking the lactate, we are taking the electrons away from it, we're turning it back into pyruvate, and we're creating more NADH, because remember that balance between NAD+ and NADH has been restored again. So if the electron transport chain is working regularly, then it would be a good thing that we're trying to produce more NADH. So we're just taking those electrons away from lactate, putting them back on NAD+, so we now have pyruvate and NADH, which are two useful molecules. And we can use those for the electron transport chain. So the pyruvate that can then enter gluconeogenesis, it can turn into glucose again. It can enter the Krebs cycle. If it goes to, if we turn it into acetyl-CoA, it can enter the Krebs cycle and spin there, or it can be sent back into the muscles. So there's a couple of things we can do with that pyruvate, but it's just important to know that the lactic acid, although it's considered a byproduct for anaerobic fermentation, it actually does get repurposed once it goes into the liver, and that process is known as the Cori cycle. Okay, and this brings us to our last review question. So again, take a minute, read it over, and we will go over it. So which of the following molecules is or are involved in fermentation? We have Roman numerals 1 through 4, and then our choices A through E. So the correct answer is D. This is a pretty simple question. You basically, I mean, you don't really even have to have extensive knowledge of fermentation to know which of the following molecules is or are involved in fermentation. One is fructose, which I think the only time I mentioned fructose was probably in the beginning of glycolysis, so it's not really important for fermentation. But pyruvate definitely is, lactate definitely is, and ethanol is as well, because remember, we have lactic acid fermentation. Lactate and lactic acid are basically the same thing. And we also have ethanol fermentation, so those two definitely are involved, and then pyruvate, of course, is involved as well. So the only one that's not is 1, fructose. So that would make D, 2, 3, and 4 the correct answer, and A, B, and E incorrect.